It's a real pleasure for me to introduce our final speaker of the day, Dr. Larry Corey. He's the Senior Vice President of the Fred Hutchinson Cancer Research Center. He's also the co-director of the vaccine and infectious disease program there, and the head of the ID and virology programs. And this apparently leaves him with plenty of free time because he also runs a research program that is pretty remarkable in its breadth. Spanning, I guess, epidemiology, immunology, and vaccine development for numerous viruses, including herpes simplex virus, HHV-8, and HIV. Just one sort of small example of this is, I guess now 20-some years ago, he made the discovery that, or the observation, I guess, that there's this interesting correlation or strong correlation between genital herpes infection and susceptibility to HIV. And so you'd think that then just treating people with drugs like acyclovir would decrease that susceptibility, but in fact they found that it has no positive effect at all on that. And he's gone on to show that in fact these herpes outbreaks cause this sustained inflammatory response at the site of the lesion after the lesion has regressed, and that a component of that is are two cell types that are really integral for establishing an HIV infection, which would be CD4 cells, T cells, and dendritic cells. And so I guess I've heard him refer to this as creating sort of the perfect storm for an HIV infection. And this research is really important in that I think it highlights the strong need for vaccines against genital herpes, which is something that, of course, his group is actively working on along with many, many other things. He's won numerous awards, which I won't go into, and like the rest of the speakers today is, I think, just generally awesome and should serve as a real inspiration to the rest of us. So delighted to have you here today, and welcome.
That's a sweet introduction. And now I'll always say, and my wife says I take out the garbage loudly. so she's grounding. All right. Well, you've heard most of the talk, so no. So I actually will title this sort of talk as a 30-year quest of missed stumbles. So I'm going to take you through the story of the pathogenesis of herpes, and I think it's an interesting story. Maybe lessons in self-awareness and self-discovery, and to not always believe the original crap that you write. And when you start believing it, you're no longer a good investigator. And yeah, I'll get this back. There we go. Does that work? Okay. It's off. All right, so let's just start. So there are currently 8 human herpes viruses. The alpha herpes viruses are HSV-1, 2, and varicella zoster. The beta herpes viruses are CMV, HHV-6, and HHV-7, and the gamma herpes viruses are KSHV and EBV. The reason I actually even go through this it relates to the issue of latency and the role that latency plays in the pathology of this disease. So let's get that over there. It's off, but we'll put it over there anyway. They're all large double-stranded viruses. They look a lot. We're not going to look by EM a lot. We're not really going to talk a lot about the epitope aspects of vaccine design. I really want to concentrate on the pathogenesis of infection. But one of the important aspects of herpes viruses is the structure of the tegument, which actually seems to be a target for much of the CD8 T-cell responses, and we'll talk about the importance of that in a minute. Latency, as you all know, is partial gene expression, and the mechanisms differ in each herpes virus regarding the number of genes expressed and the structure of the latent genome. And really, when you sort of look at the conceptual framework of herpes virus infections, or maybe any other latent virus infections, high titer persistent viral infection is probably the prototype is felt to be HIV, whether you want to think of EBV that way. HSV-1 and HSV-2 and VZV, the alpha herpes viruses, have standardly been thought as infections in which you get an acute infection and you establish really tight control of latency in the dorsal nerve root ganglion. And if you actually look at the chapters I've written in herpes for the first 20 years of my life, and listen to my lectures, I would really start this, well, the virus is latent in the neuron, sipping a core, is having a good time, and only intermittently reactivates. And I'm going to just tell you that is wrong, 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 wrong. And that's, really the revelation of how you miss the underlying biology of the disease over time. The classic smoldering infections have been CMV and EBV and HHV6, in which, we really sort of can essentially almost always chronically, somewhere in the body, persistently isolate the virus. And so latency sometimes in your own mind has to be, differentiated on an organism level and on a cellular level, and also in a spatial level. And I think that's really one of the things, one of the messages out of the talk. Now, the acquisition of HSV-1 or HSV-2 is really contact with infected secretions. It's well known for 30 years, 40 years that people asymptomatically shed the virus in either their saliva or genital secretions. What has not been appreciated is actually, as I'll talk to you, is the frequency of that. The initial mucosal infection gives entry into the sensory nerve root. There's anterograde spread into the dorsal nerve root ganglia. There's replication locally, both contiguously in the dermatome and across. There's centrifugal migration with the neuron to the epidermal area. There's a release of virions from the neurons. There's infection of epithelial cells and skin lesions or subclinical shedding. And the standard dogma has been that when you have 3 or 4 or 5 days of epithelial shedding or genital lesion, it's due to contiguous cell-to-cell spread by the epithelial cell. And I'm going to actually show to you that that's actually just really, there is contiguous spread, but this is actually a multifocal disease. And when you get 3 or 4 days in a row of viral shedding, it actually is from multiple seeded sites from spatially different areas. And we'll talk about that and the implications of that. Now, we'll continue the first part of herpes 101. HSV can develop latency and reactivation in the trigeminal nerve root ganglion. For HSV-1, that's usually from getting kissed early in life, and it can be in the maxillary mandibular or ophthalmic division. If it reactivates in the eye, you get herpetic keratitis. It reactivates in the lip, you get a cold sore. About 90% of the lesions are, 90% of infections are acquired asymptomatically. About 8 or 10% give you a minor illness, and about 1% actually cause disease, which can be either gingivostomatitis, of the mouth, keratitis, genital herpes is caused by HSV-1, and then the more other forms of the disease, encephalitis, occasionally with primary infection, but usually with reactivation infection. Herpetic Whitlow is of the eye, and herpes cladomatorium is from wrestling, and you can stay away from that one. HSV gingivostomatitis is this classical presentation in the child. HSV keratitis, sort of this classic dendritic ulcer of the eye. Primary genital herpes, the analogous situation with gingivostomatitis gives you an exocervicitis, very classic genital lesions, and reactivation in the buttock. Neonatal herpes is the most significant direct complication of genital herpes on the West Coast. About 1 in 3,500 live births are of neonatal herpes untreated. It's the most deadly of any neonatal disease. It's not a disease that we've actually decreased since antivirals. It's a little weird that we actually don't have a public health control program for neonatal herpes. It is the infectious disease untreated with the highest mortality, and even with treatment, we have a very high morbidity for this disease. It's relatively uncommon in any obstetrician's personal life, but on an epidemiological basis, it is really the childhood infection that is most from which we do not have a public health control. Now, the most significant indirect complication of HSV-2 infection is HIV infection, and we'll talk about that. So what is the epidemiology of HSV worldwide? And just say that it is a disease that is pandemic throughout the world. In other words, I'm telling you, no place is safe for sex, maybe a little part of Spain. This is HSV prevalence among women, mainly from antenatal clinics. By definition, they're sexually active if they're pregnant. So the prevalence in the United States is 16 to 20%. And if you'll see in Africa, I'll show you some data where the prevalence is 50, 60, 70%. And frankly, there's been no place in which there's a sexually transmitted HIV epidemic in which the HSV-2 epidemic has not preceded that or anteceded that, whether that's on a countrywide basis or in a closed population basis. And we'll come to give you a sense of in sub-Saharan Africa what the HSV-2 rates are is, they are anywhere from 60 to 80%. Now, you always see that women will have higher prevalences than men, and that's characteristic in every population, anywhere from a good 5 to 20%. But you can just read down the countries, and you can see that in almost all countries the rates are very, very high. Rates are lower in Islamic countries and in the Middle East, but they are in select populations starting to increase. In high-risk groups, the rates are astounding. In commercial sex workers throughout Africa, the rates are 80 to 90%. Now, as I take over as president of the Hutch, I hope this curve will be our endowment, but it is not. It is not at the moment. It is General Herpes office visits between 1966 and 2008. And this is really a Will Rogers effect. This is actually more an observation effect. There have been, or a case detection and awareness effect. There have been serosurveys in the United States. I'll show you the data in a minute that show essentially the prevalence is pretty stable. In fact, it's maybe gone down by 1 or 2%. So the increase in case recognition relates to the availability of treatment, the increase in awareness, and the increasing availability of diagnostic assays. Now, there have been nationwide surveys of HSV-2 in the United States. HSV-2 is probably the best biomarker we have overall of sexual activity. Albeit I'll show you that that it's not just the number of sexual partners, it's your sexual network that determines your risk and your seroprevalence. So overall in the United States, it's 16% of the population. As you can see, it's significantly higher in women and men. Like all chronic diseases, it has a cohort effect. So you can see increasing prevalence with age. And again, you will see women always at higher rates than men. If you look at ethnicity in the white population, it's around 20% in women. In the black population in the United States, it's 50% of the women, and Hispanics also around 20%. Like all sexually transmitted infections, the number of sexual partners are related, but you can see that at similar sexual partners, there are very, very different seroprevalence rates between Caucasians and African-American populations. And again, it relates to once it penetrates of high prevalence into a cohort, it is that continued high prevalence. It's serial monogamy that this is a disease in which the epidemiology is already in the general population, and with serial monogamy, one sees increasing incidence. The two serosurveys that have been published in the United States, one between 1999 and 2004, and 2005 and 2008. This is about the same seroprevalence. 87%, in other words, you ask the question of HSV-2, people who are HSV-2 seropositive, they ask the question, do you have genital herpes? Then they look at the percent that subsequently are HSV-2 positive. There's a large iceberg phenomenon. 82% to 87% of people have unrecognized infection. Ergo, we would expect nothing other than any control of this infection until we develop a vaccine. So how about HSV as a risk factor for HIV acquisition? What's the epidemiology of that? Well, HSV-2 was really part of the original description of HIV. In the original description of HIV, two of the patients had prolonged HSV-2 infections as part of their clinical manifestation. So it was known to be, of course, immunosuppressed patients have problems with mucosal HSV infections. In 1988, Walt Stam and I published the first data that we were following a cohort of gay men, and we actually showed that both incident and prevalent HIV infection was associated with HSV-2, both prevalence and incidence. Since that time, 31 studies and 22 manuscripts have essentially shown an increased risk of between HSV-2, not defined by genital ulcer disease, but by antibody and increase of HIV. In men, it has been about threefold, in women about threefold, and in MSM, the relative risk overall has been 1.7-fold, although in the STEP study, we actually looked at this, it was actually threefold. So in the United States, in the STEP study, as part of the vaccine, that was similar between the vaccine and the placebo group. But it is definitely an increased risk in HIV acquisition. And this is a meta-analysis from Anna Wald, who has worked in our group. This is in developing countries and developed countries, in men and women. All the relative risks are always to the right, just to say that there's a huge consistency in the data showing this association between HSV-2 and HIV. One of the more interesting studies was done in Africa, in which they looked at discordant couples in 4 separate cities against 4 different, very different demography and sexual behavior of 221 couples where one partner was HIV positive. There was a 51% HIV positive concordant versus 49% HIV discordant. And HSV-2 concordance was the strongest predictor of HIV concordance, odds ratio of 9 and 4. Now, on a population attributable risk, if you actually look at the prevalence of HSV-2 and look at the population attributable risk in the United States, the population attributable risk is estimated to be around 30%. If you look at MSN and in African American men and in sub-Saharan Africa, where the prevalence rates are 50%, the population attributable risk may be as high as 60%. And in sex workers in high-risk areas where the prevalence of HSV-2 may be as high as 80%, the mathematical modelers say that the population attributable risk may be as high as 70%. I'm not quite sure it's quite like that, but it makes for a good talk. Now, the biological plausibility. Now, the biological plausibility has always been there in the sense that we've known for a long time when we first biopsied HSV ulcers that the second cell in was the CD4 cell. So we always felt that the pathogenesis of this was that it causes macro and microscopic ulcerations, brought CD4 cells that were activated to the surface, and that was the biological plausibility. Now, the problem with that people argued over this for so many years, frankly, not really believing all the epidemiological data, was that, herpes just didn't reactivate, just wasn't around long enough to actually account for, this major factor. And so, we started doing studies about the fact that, well, when we started looking at herpes, there was a lot of asymptomatic shedding, and we sort of took that biological plausibility that from saying, oh, this is a virus that reactivates once a month to a virus that reactivates 10 to 15% of the days. But we still were really thinking that, that its mechanism of action was really that that it was the organism that was causing the ulceration and was the break in the epithelial barrier that was the cause of this. And we essentially reasoned that we had an antiviral drug, a series of antiviral drugs that we had had for a long period of time, acyclovir, valacyclovir, that were very effective clinically in preventing the both asymptomatic shedding and clinical ulceration. 80% effective. And therefore, if we shut down that viral-induced injury, that we would essentially protect the epithelium and reduce the acquisition of HIV. And we embarked upon a series of placebo-controlled trials to look at that. And these have been published and alluded to, and they were very, very well done trials by led by Connie Kellum and Jay Langappa from the University of Washington, and looking at heterosexual men and women throughout both the Americas and Africa, putting them on acyclovir versus matching placebo, a dose of acyclovir that should have worked and would work. We had incredibly good compliance. And we got this depressing Kaplan-Meier curve. We had incredible adherence. So it did absolutely nothing. And it did reduce genital ulcer disease, not as much as there's some interesting things out of Africa, whether these drugs work as well in Africa as we have in the huge numbers of studies that we've done in the United States and Europe with acyclovir. After all, it's a drug that is used like water, gazillions of kilograms made, sold per year. So no effect on HIV acquisition. It reduced genital ulcer disease, and the failure was not related to adherence, resistance, absorption. It was, just dead negative. You had the wrong, idea here. So what was wrong? So what went wrong? And this is me here who did all this stuff, And so I think out of ignorance, we avoided, we ignored the first axiom of infectious disease, which is never underestimate your pathogen. And we went back to basics about what's the underlying pathogenesis of HSV reactivation. And we had to really challenge our models of what the model was of reactivation. So the model of reactivations, for the last 50 years has been essentially static models in which HSV release from latency was felt to be relatively infrequent, or even if it was, even relatively frequent, it was not all the time, and was under tight control. And, all of the molecular virology of HSV has been fixated on people studying, host neuronal interactions, what's going on with the virus in the host cell. And the concept that the immune system was actually the dominant force in controlling reactivation was really not in the, even considered. Now, some work started being done by Bob McKendrick in mice about 7, 8 years ago to start saying, well, maybe the immune system actually controlled virus in the ganglion, but the fixation has always been on the ganglion. So I'm going to show you a series of data that says this is all wrong. That HSV reactivation in the genital mucosa is really a constant phenomenon. That the virus reacts on average at least every 7 days and probably almost every day. The most frequent reactivation that we virologically identify, that was when we swab the genitalia, lasts less than 6 hours. So it's on and off. And antivirals, even in high doses, do not stop these short episodes of reactivation. They alter the long episodes. So we've been deluded about how good these antivirals are because we were studying just the right-hand side of the bell-shaped curve. And we were very effective in doing that. But on the left-hand side, I'm going to show you some studies we've done. We give huge doses of acyclovir. They don't do anything in shutting off these short episodes of viral shedding. So they're not biologically effective. They're clinically effective. Also going to show you some data that the mucosal immune system contains almost all the reactivations. In fact, the most common reactivation is one in which the mucosal immune system actually controls the virus without it ever even causing a reactivation. And I'll show you some biopsy studies that we have clear proof of that. In fact, this is actually the most common thing. So the virus and the host are interacting in staccato time. And subclinical reactivations in which we identify herpes virions on the mucosal surface constitute approximately 40% of clinical reactivations, less than 10% of the total episodes of HSV-2 into the periphery. So in other words, the host and the virus are sort of seeing each other and battling this out on a nearly daily basis. This mucosal reactivation is associated with a chronic persistent inflammatory response. And this inflammatory response occurs despite normal appearing skin. And the inflammatory response, as you have heard, consists of HIV receptive positive cells. These are CCR5 bearing CD4 cells and DC cells expecting the lectin DC sign, thus explaining this interaction between HSV-2 and HIV. So there's a disconnect between the underlying biology and the clinical manifestations of HSV. It's really, since the HIV epidemic has really felt to be, a trivial mucosal disease. It's sort of been the Rodney Dangerfield of the virological field. But I'm telling you, the biology is underappreciated, and hence and that's why we've seen, despite all the antivirals, essentially, these are case management tools, but they are not affecting the underlying population epidemiology, and more importantly, essentially the population biology of this disease. And we need to think more about that. So let's go through the data. So we used a high-tech thing, people swabbing themselves. I mean, what else do you do in Seattle when it rains? This is just nothing but a Western blot saying we take HSV-2 positive people and we give them this packet of stuff, of which we tell them swab yourself. We teach them how to do this. We keep these diary cards. And we started doing this years ago actually found out that the rates of acquisition and that people actually take better swabs than clinicians. And that this is not so high tech. They do it, we put it in a PCR buffer, we do a wonderful quantitative PCR data, and it's amazingly sensitive and specific. So actually, the first insight that we had we were amiss came from what I talked about with a mathematical model that suggests that when we had 3 or 4 days of shedding, when you looked at the pattern of the shedding, the Markov model said that when you had 3 or 4 days, it was more likely that it was 2 separate episodes rather than one contiguous episode. So we said, well, let's start looking more frequently. So Karen Mark, who's now at the Berkeley Health Department, and Anna started a study in which we started culturing people 4 times a day, every 4 to 6 hours. And lo and behold, we found that, 46% of the episodes lasted less than 6 hours. Almost 55% lasted less than 12 hours, and a smattering over here. So the most common episodes were really these short reactivations. There was a very nice association between, and we did this with oral disease also. There was a very nice association between the titer of virus and the duration. So when you shed higher titers, you had a longer duration of the episode. Now, this helped explain a study that we had gotten a lot of credit for, but we're really quite disappointed about, that we published in 2004, which was a study in which we used valacyclovir, very similar to the IPREX study. It was sort of the first concept that you could treat someone with a chronic viral infection. We put the people who were HSV-2 positive, who were in a monogamous sexual relationship with someone, and gave them valacyclovir and looked at whether it reduced transmission of HSV-2 in the sexual partner. And we showed it did it by 50%. Now, and that's what this Kaplan-Meier is showing. But we were actually quite disappointed with that. Frankly, in the sense that when we gave HSV-2 to that partner, who was HSV-2 positive, we shut down viral shedding when we were culturing them once a day by 80 or 85%. It was like, well, wait a minute here. Why are we shutting down virus by 85%, but we're only decreasing the biology of transmission by 50%? So then when we had these data, the light bulb went on saying, there must be these short episodes that we are not necessarily treating that we have never picked up before because we're culturing only once a day. And I've just shown you when we start culturing 4 times a day, hey, we get a different answer. So we went about, Karen went about actually doing that. And so we took a study in which we took people on the standard dose of valacyclovir that we used in this transmission study. We then used a high dose of acyclovir to see if more would be better. And we started culturing them 4 times a day. And we switched them over and we analyzed the data. And lo and behold, 5% of the swabs, they broke through with these short episodes in 9% of the days. So even at very high doses, you're not, and the duration of these episodes was a median of 6 or 7 hours. So the antivirals were clearly not stopping these short episodes. So each person averaged around 3 episodes. They were low copy episodes, but obviously low copy is still able to transmit the disease. So even on therapy, the person averaged 1 episode per 33 days. So we think this sort of explains, and if you actually look at high titer episodes, they're very infrequent. So we think it explains these results. We then have recently decided to up the ante, and we actually gave very high doses, valacyclovir 1 gram TID. So this is the zoster dose, about as much as you can. And it still breaks through. It reduces this from once every 33 days to once every 45 days. But again, this class of drugs in which you actually have to, they're wonderful for their specificity, in which you have to have it's phosphorylated by a viral specific enzyme. But you need to, the virus is in the cell, so it's the race between that and inhibition, and it actually doesn't do that. It doesn't affect release of virions from the neuron into the epithelial cell, and it's actually not affecting the underlying biology and pathogenesis. And I'm going to show you some slides that it doesn't affect at all the chronic inflammatory infiltrate that occurs with this infection. So frequent short bursts of HSV-2 reactivation occur in the presence of both standard and high-dose acyclovir and valacyclovir. And I think these observations likely explain the discrepancy between the success of suppressive therapy on an individual basis to prevent clinical recurrences, but the failure of suppressive therapy to provide better protection from either HSV transmission or HIV acquisition. So there's plenty of room for improvement in HSV therapy. So let's talk a little bit more about these reactivations and anatomically from whence do they occur. If you look at all the chapters I've written in Harrison and Mandel and everything about herpes, I really talked about reactivation being unilateral and unifocal. It was felt because of, primary infection was bilateral and established latency bilaterally and did up and down the dermatome, as I told you. But once it was contained in the neuron, the concept was that, one neuron on the right side, neuron on the left side, they're not connected. You would just sort of reactivate on the right side, and you'd get a unilateral and unifocal reactivation. And clinically, that's the way it looked, and it looks, and pathophysiologically, that's what we and I believed. And that is wrong. So when we started looking at this issue of looking at, well, from whence is all this multiple day shedding occurring every 4 to 6 hours, we started saying, well, let's do some, physician-collected swabs. Let's break the genitalia into 7 or 8 areas, and we'll do 7 or 8 swabs a day. We thought that was a lot, and that we'd sort of look at this. And so our first look at it was, lo and behold, we actually started getting swabs from the right side and the left side, positive on nearly every day. And it was like, okay, the lesion might be down here, but we were picking up shedding on the left majora and the right majora, on the left side, the right side. These were pretty high titer. They were all over the place. It was like, geez, 80% of days had bilateral shedding. Is this cross-contamination or what? What's going on? So we decided, hey, we're going to GPS map the genitalia. We're going to do it better. And at the same time, so now we're collecting 22 swabs a day. And we also developed this rapid PCR assay that we were using for the delivery room, in which we could get an answer in a 2 or 3 hour period of time. We don't need 2 or 3 hours. We sort of did it in a 24-hour period. So we took a group of people who would come in every day, and they get 22 separate swabs. We'd run the PCR on them. And if it was positive, and we'd have the answer by late afternoon, evening, it was positive, we'd bring them back the next day, and we'd biopsy the site in which it was positive. So that's the studies that you're going to look at. So from whence looking at the shedding pattern? So here's this is one that sort of is classical, sort of the red means you're positive on a day, and I'll lead you through the anatomic area, and the pink, as you get hotter, this is the amount of virus. So low titers are in the pink, and red titers are in hot, and so this is, okay, one area on the left side and one on the left side, right side. This is sort of the classic case, the way it's, way the textbook have it. But, here you have this reactivation, multifocal on one day. And then it goes away for a couple days, then unifocal. And then you have this in which it starts here, and then the next day there's a huge amount. There is a genital lesion at this period of time. It's rapidly cleared. Here's on one day unifocal on the left side. Here's on the right side. Here's again a day up here and down here and over here. So these are very widely spaced in the genitalia. And we could go through this, but trust me more, that's the interpretation. We've actually done a mathematical model in which we've actually looked at the spatial association. Does detection of HSV at one site impact the detection of a neighboring site? And the answer is, this is what the data look like. No. So we don't really think this is cross-contamination. So reactivation is multifocal. Does it emanate from frequent release of virions from neurons at multiple sites? Herpes is in the vagus nerve. The sympathetic and parasympathetic systems have a more important role than we appreciate. Varicella zoster has been isolated in hair follicles. Are we actually seeing, again, the autonomic nervous system playing more of a role than we've actually thought about? Is this contiguous spread of epithelial cell virus with much more rapid than appreciated, or both? And I think the answer will probably be a little bit of both. So what's the pathophysiology of these reactivations? Again, what do the biopsies show? So the biopsies are showing clearly an inflammatory response. So here's a biopsy from this area. Now, first of all, you see no ulcer. Now, it's possible that there is an ulcer. I mean, we biopsy an area, it's still bigger than a genital lesion. So we could be missing some things. But, clearly right here is a focal inflammatory response. If you actually stain that for CD4 and CD8, this is a CD8 stain, you see a focal cluster of CD8 cells. If you actually take those cells and look at whether they're herpes reactive, we actually, at least for CD4 cells, are shown that about 10 to 15% of these cells are HSV-specific CD4 cells. So, it's pretty clear that we are seeing an immune response and a herpes-specific immune response in these areas of asymptomatic reactivation. So the next question is, how does the host contain reactivation so quickly? I mean, let's think about it. We've actually started doing some studies in which we look at people every 2 hours. And we see shedding episodes for 2 hours. And this is like 10 to the 4th, 10 to the 5th virus. And then 2 hours later, it's gone. So how do you mobilize an immune response on a mucosal surface this quickly? What is controlling this? So I'm going to show you that the mucosal immune reactivation is associated with a chronic persistent inflammatory response in the dermis. We've already talked about what that inflammatory response is. And so it is really the mucosal immune system that is sort of fighting with this infection every day. So the study design that we did on this was one of, a sense of heroic patient participation. Because first of all, despite what they're doing here, which is going through these sequential biopsies, these people also were in study with us for over a year and a half, because no one had defined the HSV-specific T-cell responses. And we needed to make reagents that were epitope-specific. So we actually had to bleed the people, do epitope mapping, define what their CD8 responses are for the specific HLA types, make some unique reagents, and then biopsy them for these lesions, and then do all the studies that we're doing. Now, sometimes we didn't have to do that. For the CD4, CD8 studies, the reagents are the reagents. But for the studies in which we're actually looking at HSV-specific tetramers, we had to develop the reagents. So we did a study in which we took these people who are symptomatic in herpes. When they had a lesion, we biopsied the lesion at the time it was healed, and then at 2, 4, and 8 weeks later. We actually then waited for another lesion, usually a month or 2 later. We then put them on chronic acyclovir, and we biopsied the lesion and did the same thing. And when we biopsy them, it's not like we biopsy that area at, let's say, 12 o'clock, and the next one we do at 3 o'clock, and at 6 o'clock, and at 9 o'clock. So, we're circling the lesions. We've done a lot of these over time. Lo and behold, when we did this, we found, first of all, when you have a genital lesion, there's a huge inflammatory response. And this is CD4 cells per millimeter skin. This is weak. But, and we'd also biopsy the contralateral genital area. So the opposite side, which we thought at that time was actually not a bad control. Now, that's before we actually looked at the multifocal stuff. We're now sort of saying, I'm not sure this is such a great control, so we're now looking at the arm. So the data, the arm's really clean, but this is really as good, a tough test of the response. It gives you a sense that this is truly a persistent inflammatory disease. This is CD8 cells. This is CD4 cells. This is how it looks. Just an enormous inflammatory infiltrate. We then wanted to ask, well, are there herpes-specific cells there? And for that, we had to develop a new class of reagents. In situ tetramer staining works on lymph nodes. Stuff that Ashley Haas does, but in skin there's a lot of autofluorescence, and the tetramers with strep avidin don't work, and so we developed these fluorescent tetramers with these fluorescent-linked quantum dots that really are about 100 times more sensitive in skin. And lo and behold, when we did this, we found this persistence of HSV-specific CD8 T-cells right at the dermal epidermal junction. That persisted for long periods of time. So this is the epidermis. This is a reed plug. This is right at the dermal epidermal junction. This is the sign for CD8, and this is the B7 cell. So the yellow is actually is essentially a tetramer staining of an HSV-specific CD8 cell. And if you actually look at this, and then we actually stain this with NCAM, which is for a neurofibrillary stain, you actually see that these CD8 cells are right at the contiguous area of the neuronal bulb. So giving just from a neuroanatomic point of view, you're like, what are you guys doing here? You must be doing something there. We'll come back to that story in a minute because we pursued that. So whether you put persons on acyclovir or not, you again got this huge inflammatory response and then persisted, and this is what the control cells, this is CD8, CD4, and you sort of again see there's 8 to 10 times more CD4 or CD8 cells at the genital herpes site and as compared to the contralateral skin. And again, to give you the sense of what this looks like, this is where it is in healing, this is in normal skin. So again, you see this enormous inflammatory response. The CD8 cells are at the dermal epidermal junction, the CD4 cells tend to be in the dermis. And again, here's 12 weeks and here's 20 weeks on acyclovir. It didn't matter difference whether you're on acyclovir or not. If you actually ask the question, tell us more about these CD4 cells, yes, they are enhanced in CCR5 expression, more in the tissue than in the blood, more in the herpes lesion than in the contralateral control, and no different whether you're on acyclovir or not. Now, what else is in there? I can't tell you everything that's in there. So one of the things that we looked at immediately were dendritic cells. Now, why did we look at that? First of all, dendritic cells give you immunological memory. I mean, you know, God made herpes way before HIV. And so why would the mucosal system want to have dendritic cells, especially plasmacytoid and mononuclear monocytoid dendritic cells in genital skin? Well, because they're great for memory and they're great for antigen presentation. And if you're fighting this organism all the time, you'd want to have them there. And that's why they are there, and that's why we stain for there. And they are there, and they persist, and acyclovir didn't make a difference. But that then creates what you know, HIV really loves. You know, it has, in about 10% of the sections, we saw this, you know, wonderful, beautiful picture of a CD4 cell contiguous to a CD123 cell, or what here is even more a CD4 cell contiguous to a DC sign bearing cell. This is red DC sign. This is CD4, and about 10% of the. So essentially the inflammatory synapse that really essentially provides the spark for HIV infection is really enhanced in this persistent viral infection. So we no longer, you know, the missed concept was that it was the infectious ulcer that was needed. You don't need an infectious ulcer. I mean, I really should have understood this in the first place. Every time you have sex, you rub two surfaces together. You create an enormous number of microulcerations every time you have sex. Whether it's vaginal sex or insertive, you know, receptive anal or insertive anal sex. And if you have a persistent inflammatory response in your genitalia, there you are. All right? So HSV-2 infection leads a persistent, previously underappreciated subclinical inflammatory response of HIV receptor positive cells in the dermis of clinically normal appearing skin and mucosa. This inflammatory response is localized to the site of prior reactivation. This inflammatory response is enriched in CCR5 expressing CD4 cells and a mixed population of dendritic cells, as well as DC sign expressing cells. These CD4 cells are often contiguous to DC sign expressing cells, and the quantity, the spatial localization, the duration, the type of inflammatory infiltrate post-healing is not influenced by antiviral therapy. We then took actually some of these biopsies and did a sort of an ex vivo experiment. We took the biopsy from a genital lesion and the contralateral site and in vivo infected it with HIV. And lo and behold, there's five times more HIV infection in the lesional as compared to the controlled biopsy. So as much as you can mimic this, you know, in an ex vivo experiment. We think this pathology likely explains the increased risk that HSV-2 has on HIV acquisition and explains why acyclovir therapy is not there. And so, you know, this is sort of a cartoon of an HIV bouncing in onto a green CD4 cell, but it's really when the CD4 cell is next to the dendritic cell to the left of here that you get the spark. Now, I want to spend 2 more minutes and then we'll get out of here. We then looked at this issue of immune surveillance. You know, can we prove that the CD8 cells that are sitting contiguous to the neuronal area, are they functioning as immune surveillance, and how do they function? And that's been the direction of the lab lately. I'll just give you a sense of how we're approaching that. We're approaching that by laser capture microscopy associated with transcriptional arrays. So we want to look at cytolytic function and non-cytolytic function. And so we are able to actually, use this issue that we know spatially where these CD8 cells are. So we developed a rapid CD8 stain. We are able to pop them out with laser capture. Just to give you the purity of this, these are the junctional CD8 cells. We get no CD4 and huge amounts of CD8. If you actually look at the whole tissue, you'll find that there's CD4 and CD8. So we have pure populations of CD8 cells. We're starting doing the transcriptional arrays. And like any time we have an unbiased thing, we're getting everything. So I mean, these are really, truly metabolically active cells. If you look at the genes associated with cytotoxicity, there's a huge number of genes that are upregulated, both perforin, granzymes, as well as chemokines and cytokines and proliferation assays. But we have gone after the cytotoxic genes, and we've actually shown not only on a transcriptional level, but on a protein level, that we can, here's in a biopsy in which we see HSV-2 antigen in red. This is actually a CD8 cell sitting right next to an HSV-infected epithelial cell from one of these asymptomatic biopsies, and this CD8 cell expressing both granzyme B and perforin. So, we really do feel that, hey, and this is another slide showing the same thing. So, and I'll just say that, we have 1,000 genes. Activation, it's one of those things, so like, what are we going to do with all these things? And how are we going to analyze, are there some unique chemokines or non-cytolytic mechanisms that are really being associated with reactivation and containment or not? And that's what the obsession is in the lab. So the last thing I want to say is just give you a 2 cents of wanting to look at the issue of, is it the neuron or is it the host? And in the human, we're never going to be able to biopsy the neurons. So how do we solve for what's going on at the neuronal level as far as it releasing into the skin? And I had an incredibly bright fellow who was mathematically inclined. We sent him to the London School to do mathematical modeling, and he's done this. He's now come back and done these intrahost models of HSV pathogenesis. He's published 2, Josh Schiffer, he's published 2 wonderful papers on the mathematical model. And just to say that what the mathematical is saying is the following, because I'm running late. At least I think I'm running late. 2 minutes, okay. The mathematical model says there are 3 types of reactivations: baby bear, mama bear, and papa bear, okay? Now, baby bear is 85%. So this is a typical person in a year. This typical person, and this model is now on 365, it's a huge database of 365 people that we've followed with over 14,000 recurrences on these daily shedding studies. So this is a robust database. And so number one is that about 80% of the episodes are mama bear, are baby bear episodes. The baby bear episodes, really have very low number of infected epithelial cells, anywhere from 1 to 100, and are rapidly contained, average only 2 logs. The mama bear episodes, about 10 or 15%, average around 180 infected cells, 180 peak infected cells, so about 8,000 infected cells per episode, and that gives you a barely discernible peak diameter. These are the episodes that are contained in the mucosa, the baby bear, that actually never cause any ulceration whatsoever. And it is only the papa bear episodes that have a huge number of viral infected cells that actually are the clinically expressive episodes. And the difference between this, and this models actually the frequency of reactivation and the average lifespan of an infected cell and the amount of HSV DNA per episode. And the most important aspect of this is that the difference, if you actually look at the line here, to get a papa bear episode, the duration of a viral infected cell is 90 minutes. To get a baby bear episode, it's 30 minutes. So you're actually talking about the necessity on an average basis of essentially getting rid of a viral infected cell within an hour. If you don't get it in an hour, that's the difference between containment. Every 20 minutes, focus you on the difference. I don't think HSV is actually different than HIV. I think the time span in which we think of viral host interactions are not actually the real time span that they actually occur in vivo. We do our experiments the next day or the next 2 days, not in the next 20 minutes or 30 minutes. We are not thinking about it in the terms that are actually biologically occurring. And I think that's really one of the sort of most interesting things in the model. The last thing I'll say is that the model actually shows that CD8 T-cell density actually influences the infected lifespan, and there's a threshold effect. And if we get above this threshold of CD8 T-cell density, we actually can contain. They're only baby bear episodes. When that threshold goes down, there's a mama bear or papa bear episode that's associated with replenishment and then containment. and then it goes down in replenishment and containment. And actually, that goes with the transcriptional array. We are actually not seeing PD-1, or we're not seeing exhaustion. We're not seeing the kind of profile that you actually see with HIV. So there is some turnover. There is, there is migration in and out of, and so we're starting to cut out the CD8 cells and sort of look at, sequencing, 454 sequencing to sort of look at what we're getting out of there. But so when you look at things like lesion diameter, infected cell lifespan, and I'll show you reproductive rate, there's this wonderful correlation between, this is high copy episodes and low copy, an inverse correlation between CD8 lymphocyte density and the lesion diameter, the infected cell lifespan, the reproductive number, all the things that, that you would expect. So, and if we look at the model and sort of say, well, we're going to flood the model with more and more neurons, if we sort of say we increase the number of neurons released in the potential model from 100 to 100,000, it never affects the reproductive number of lesion size. What affects it are all things associated with the host immunity. So I'll just end with that to sort of say the major message is that HSV is not a static intermittent infection. It's a persistent, rapidly cleared mucosal infection, essentially producing a chronic inflammatory response in the entire genital tract. And immunotherapeutic vaccines directed at prolonging the number of or function of host responses in the periphery may be a potential novel approach to reducing reactivation and reducing this chronic reactivation process. And so the old model of HSV pathogenesis is not true. The new model is really, it's occurring all the time, all these interactions. Papa bear, mama bear, but lots of baby bears. And so I would say you don't do this kind of work without a lot of people. And I've been very lucky throughout my life. Anna Wald, who runs the clinic and follows the patients and gets them enrolled in clinical protocols. David Cole, who's been with me and now is taking over the major intellectual thrust of the immunology lab. Ely Wang in the molecular diagnostics lab, who pump out the 50,000 PCRs we do in herpes a year. Xia Zhu and Tao Peng, who have done who developed all these nanoparticles, did all the biopsies, did all the counting, and did the transcriptional arrays. And Rhoda Morrow, who developed the HSV-2 serology, and a lot of other people. I single out Josh Sheffer and Christine Johnson, who worked in this. So thank you for your indulgence, and we hope you kept you interested.
I'm just wondering, conceptually, if it's fair to think of herpes or any virus as normal flora.
Well, to some extent, we do have normal flora. HHV6, all the herpes viruses are essentially, ubiquitous normal flora. So I do really believe that there are, normal flora for viruses. Now, that doesn't mean that normal has no biological in vivo effect, and I think that's the point we're making. So, I think we tend to think about the clinical manifestations of the disease versus the actual biological manifestation. Maybe it's not that much different than TB with respect to the issue of latency. When you have latency and persistence, there's a different metabolism, or maybe there's not a different metabolism. I actually telling you, I think in HSV it's not a different metabolism. I'm wondering in TB how much it is a different metabolism, that it is a replicating issue. Now, there's all sorts of interactions that are going on, and the speed of those interactions, I think, are being underestimated. And I think that's really one of the issues here.
I have two quick questions. One is, are your findings with the inflammation applicable to other STDs and chronically shedding viruses, things like HPV, or is this a simplex-specific phenomenon?
I don't know. We're a pretty small lab, diverted in other things, and so we don't really. We have not ventured forth. This is tough enough. Laser capture to do it this pure. It took 9 months to develop. So it's you peck away at it, as best you can.
My other question is, so I guess in my textbook, thinking of how a reactivation event from a neuron, you think, okay, it reactivates, the neuron dies, you have an epithelial infection, and then you infect a new neuron. Well, with these continual reactivation events every few hours like you're talking about, do you think that you're having just that the neurons are dying, or that just with a low level of secretion, then they're okay, and it's not like you have to reinfect and have sort of massive neuronal death all the time?
I've learned to bite my tongue as to what I think, because I've been wrong so often. And you know, I have a hard time believing the neuron dies, and I'm more of a ascriber that, you know, the glycoproteins get packaged in the one pathway and the capsids in another, and they butt out at the end, and that's when you get the infectious virion. And I sort of ascribe to that theory because, but you know, is it possible that the end of the neuron dies and there's regeneration of the neuronal bulbs much more frequently than we give credit to be? And a lot of regenerative medicine is actually suggesting that, hey, again, things in the body are not, you know, as static as we think. So I don't think it's out of the realm of possibility. Now, is the neuron in the ganglion dying? No. We don't have anesthesia of our lip, right, or you know, that occurs with HSV. But is it possible that there is some cytolytic part to the neuron and then it gets regenerated? Maybe. I don't really think that. I actually think that must be a protected space, and I think it's spatially anatomically protected, i.e. But this obsession that things are going on, that the major point of control is at the ganglion, that doesn't mean if you couldn't excise this out of a neuron, we wouldn't get rid of latency. But the obsession that things are controlled by the host neuron interaction in the sensory nerve root ganglion, you know, everybody who's funded in that area hates me for these data. But I actually don't think that that's the point. I think, yeah, latency is there, but it's not an important part of the everyday life of the virus. Just like HIV, latency is there, but it's really not an important part of the everyday life of the virus.
So I got a question up here. You said right at the very end that you have looked at the T cells and you don't see that they're expressing PD-1 or that they seem to be exhausted, which maybe is a little surprising given a constant antigen stimulation and inflammation. And so one thing I was wondering, now that you can find all of these very short and frequent reactivations, I'm wondering if you've seen any difference in the time post-acquisition of the initial infection. Do they happen more frequently later on in life, or?
Well, the data we have, which is not great data, it's cross-sectional data, suggests that people who have the disease for 10 or 15 years reactivate about 30% less frequently in our once-a-day sampling methods. Now, is it possible that they actually reactivate just as frequently, but they're shorter? You know, it's sort of like you got to redo all this stuff, and you actually don't really know what the answer is until you actually do it. So if you look at what we've actually written that, you know, that there is, and we've published a number of papers actually to say that on a cross-sectional basis, the longer you have the disease, there is some sort of, you know, stabilization, and that the rate of set point in HIV terms is lower after 10 years than it is at 3 to 5 years, and it's certainly very high in the first 6 or 8 months after infection. That's very clear. That I'm not sure necessarily the frequency of reactivation, but the ability to, the duration of those subclinical reactivations are much longer in the first 6 months after acquisition. And that's actually true sort of with every herpes virus infection. So whether that's an adaptive response or, you know, some other kind of, that's a more generalizable phenomenon which has never been really totally explained.
In tissue culture, I mean, ganciclovir works really well. I mean, in vivo, it seems to work, you know, on the overall, you know, load works really well. But here, it, you know, doesn't work that well. Do you think it's like a protection, an anatomical protection or something like that? And do you think other drugs can get there?
No, you see what I didn't get into is, for you, and it would be interesting, is that our data really are incompatible with the time frame by which we think cell-to-cell epithelial cell replication occurs. When you actually put it in the mathematical model, it really says that the virus is moving on within a 2 or 3 hour period of time. And so really, I think what is happening is that the virus, and I think this is probably true with all envelope viruses, it's actually been shown with HIV, is that it moves from cell to cell through the inflammasome. And there's transport through the inflammasome like this. And it sends some soldiers in and takes over the cell and does its thing, but there's the scout party that goes to the next cell, to the next cell, to the next cell through the inflammasome. And that's how this is occurring. And so, and I actually think that's probably most envelope viruses are doing that. And that's about the only way, that's another insight from this model. You actually can't explain the pace of all this by classical, we're waiting for the cell to burst, and cytolysis is how it gets from the next cell to cell. That is not occurring with what we end up, seeing. So I think that's what we predict, and we're actually doing some in vivo experiments now to see if we can, in vitro experiments to see if we can model that.
I think we're going to wrap it up. We're not taking a lot of time here, but I want to make a few acknowledgments. First of all, I want to thank all the speakers for coming here and giving us such great talks. And I want to thank the judges and the speakers in the competition. And I want to thank the students who volunteered their time and had the microphones and handed out papers, et cetera. And I'd like to thank Nicole and Delose for organizing this symposium. It takes a lot of individuals to make something like this go as smoothly as it did. And of course, I want to thank all of you in the audience for your attention and participation. And I think with the time as it is, I'm going to close with that. So thank you very much.